Greetings dear viewers. In this video, I would like to explain about the adenovirus and its infections in human. I am Dr. M. Manigandan, Assistant Professor from the Department of Microbiology, Sacred Heart College, Tirupattur. Introduction. The adenoviruses belongs to the Adenoviridae family. They were first isolated in 1953 from human adenoid tissues, which gave them the name adenoviruses. Adenoviruses possesses an icosahedral capsid with the characteristic fiber proteins projecting from each vertex which help in attachment to host cells. A large family of viruses with over 100 serotypes have been identified, with at least 52 are known to cause infections in humans. Different serotypes of this adenoviruses causes have tissue tropism and are associated with specific clinical syndromes. The adenoviruses infects a wide range of vertebrates, including human, mammals, birds, reptiles, amphibians, and even fishes. In humans, adenoviruses commonly infect the respiratory tract, gastrointestinal tract, urinary tract, and conjunctiva. They are responsible for diseases such as common cold, pharyngitis, conjunctivitis, pneumonia, gastroenteritis, and hemorrhagic cystitis. Transmission occurs through respiratory droplets, through direct contact, and contaminated surfaces such as fomites, fecal oral route, contaminated water, especially from swimming pools. Most infections are mild and self-limiting, but severe diseases may occur in infants, elderly individuals, and immunocompromised patients. Adenoviruses are highly stable in the environment because they lack lipid envelope. This makes them resistant to detergents and many disinfectants. Due to their ability to efficiently deliver genes into host cells, adenoviruses are widely used as vectors in gene therapy, recombinant vaccine development, and molecular biology research. Several vaccines, including some of these developed COVID vaccines, are utilized this adenoviruses as a vector through this gene transfer technology. This helps in targeting the antigens. Adenoviruses are among the most extensively studied DNA viruses, serving as important model organism for understanding viral replication, gene regulation, and host immune responses. Structure of the adenoviruses. These adenoviruses are non-enveloped viruses, making them resistant to drying, detergents, and many environmental conditions. They possess an icosahedral, that is 20-faced sides of capsule with a diameter of approximately 70 to 90 nanometer in size. The viral capsid is composed of 252 capsomeres. The capsid structure consists of 240 hexon capsomeres forming the faces of the icosahedron and 12 penton capsomeres located at the vertices. The hexon proteins is the major structural protein and contains antigenic determinants. Each penton consists of a penton base, a fiber protein projecting outward. So the fiber protein, which has in which each virion has 12 fiber proteins, one extending from each vertex. The fiber proteins consist of a tail that is attached to the penton base, a shaft, knob domain. The knob domain binds to specific host cell receptors, initiating viral attachment. Fiber proteins determine host cell specificity, that is tissue tropism. Regarding the viral genome, it is a linear double-stranded DNA with the size range from 26 to 48 kilo base pairs. depending on the adenovirus type. The DNA contains inverted terminal repeats, that is ITRs, at both ends. A terminal protein which is covalently attached to the 5' end of the DNA and serves as the primer during viral DNA replication. The co-proteins. Viral DNA is attached with several basic core proteins, that is protein 7 that helps in packaging and condenses viral DNA, protein 5 which connects the viral DNA core to the capsid, and protein mu which further stabilizes the DNA packaging, and several other minor capsid proteins are protein 3A, protein 6, and then protein 8, protein 9. These proteins strengthen the capsid and assist in viral assembly and cell entry. Overall, the virus exhibits in icosahedral symmetry. It lacks lipid envelope, increases environmental stability. The capsid protects the viral genome from physical and chemical damages. The capsid protects the viral genome from other disinfectants and other components. The surface fibers are responsible for host cell attachment, while the penton base facilitates the virus internalization via integrins. Modes of transmission of adenoviruses. Adenoviruses spreads through multiple routes because they are highly stable and survive for prolonged periods on surfaces and in water. The first method is the respiratory droplet transmission, which spreads through coughing, sneezing, and or talking. Inhalation of these infected respiratory droplets leads to infection, common in schools, daycare centers, hospitals, military camps, and crowded places. The second mode is the direct person-to-person contact. This occurs through close physical contacts such as handshakes or carrying of an infected person. Virus can spread when contaminated hands touches the eyes, nose, or mouth. The third route is the indirect contact, that is through the fomites. Adenoviruses can survive on doorknobs, toys, towels, medical equipments, keyboards, and other surfaces. Infections occur when a person touches the contaminated objects and then touches their face. This route is especially important because adenoviruses are resistant to many common disinfectants. The fourth route is the fecal oral transmission. Virus sheds in the fecal material of infected individuals. Infections occurs through contaminated hands, food, or drinking water. This mode is commonly associated with adenoviral gastroenteritis, particularly in young children. Then the fifth mode is the waterborne transmission, spread through the contaminated swimming pools, hot tubs, or recreational water, which can cause outbreaks of pharyngoconjunctival fever or conjunctivitis. Inadequate chlorination increases the risk of a transmission through the waterborne mode. Then the sixth way of transmission, the ocular transmission, that is eye to eye contact, occurs through the contaminated fingers or towels, ophthalmic instruments, or eye drops, which is responsible for epidemic keratoconjunctivitis. This is common in eye clinics and healthcare settings if infection control measures are inadequate. And healthcare associated nosocomial transmission, and rarely by organ transplantation. These are the methods of or modes of transmission of adenovirus. Pathogenesis and replication of adenoviruses. Initially, the attachment and endocytosis takes place with adenovirus infections begins when the virus encounters susceptible epithelial cells, especially in the respiratory tract, conjunctiva, or gastrointestinal tract. The fiber NOC protein binds to specific receptors on the host cell surface. that is on the Coxsackie virus and adenovirus receptors, CAR or CD46 in some adenovirus types. After attachment, the penton base interacts with integrin receptors, and this integration triggers receptor-mediated endocytosis, allowing the virus to enter into the host cell inside an endosome. Next is the capsid disassembly, where inside the endosome, acidification causes partial disassembly of the viral capsid. Protein 6 helps rupture the endosomal membrane, allowing the partially uncoated virus to escape into the cytoplasm. The viral particle is transported along microtubules to the nucleopore complex. At the nucleopore, the capsid completely disassembles, releasing the linear double-stranded DNA into the nucleus. And the early transcription and DNA replication takes place when the viral enter into the nucleus. The host RNA polymerase II transcribes the viral early genes E1, E2, E3, and E4. So the early proteins prepare the cell for viral replication, and the viral DNA replication begins using the viral DNA polymerase. During this time, the stimulating the host cell to enter S phase, suppressing apoptosis, helping the virus evade immune response also takes place. And the late gene expression, assembly, and cell lysis takes place after DNA replication, and the late genes L1 to L5 are expressed during this time. The structural proteins are produced such as hexon, penton, and fiber proteins. These newly synthesized viral genomes are packed into newly assembled capsid within the nucleus, and then the mature virions accumulate until the infected cell undergoes lysis, releasing thousands of new viral particles. These released viruses infect neighboring cells, spreading the infection. And regarding the pathogenesis of adenoviruses, this primarily infects epithelial cells of the respiratory tract, eyes, gastrointestinal tract, and urinary tract. Viral replication causes cell death and tissue destruction, leading to inflammation. The clinical manifestation depends on the site of infection. If the infection occurs in the respiratory tract, the infected person will have common cold, pharyngitis, and pneumonia. If it causes infection in eye, conjunctivitis, epidemic keratoconjunctivitis may occurs. If the spot is gastrointestinal tract, gastroenteritis will be observed. And if the spot is urinary tract, hemorrhagic cystitis will be observed. In healthy individuals, infection is usually self-limiting, and in immunocompromised individuals, adenoviruses can disseminate and cause severe pneumonia, hepatitis, encephalitis, or multi-organ failure. Dear viewers, the laboratory diagnosis, clinical manifestations, treatment and prevention will be continued in the part 2 video. Thank you for your patient listening, and kindly support us by subscribing to our channel. Thank you again.